Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Welcome to the debate.
Good to be here.
I want you to picture a facility, maybe a $500 million build. Inside, you have this stainless steel cathedral, a bioreactor holding 20,000 liters of a high-value biologic, maybe a monoclonal antibody or some next-gen enzyme. The metabolic hum is just perfect. The cells are happy, and then the invisible enemy strikes, a microscopic contamination. A foreign bacteria gets in, it multiplies, and well, within hours, that entire 20,000-liter batch is basically toxic waste.
Yeah, and let's be clear about the fallout there. That isn't just a bad day at the office. We're talking millions of dollars in raw materials, weeks of downtime for a full decontamination, and a root cause investigation that can paralyze an entire company.
Exactly. It's the absolute nightmare scenario. And for decades, our response has been, a scorched earth policy. Dump the product, blast the equipment with harsh chemicals, and start over. But what we're arguing today is that the industry is ready for a paradigm shift. So the real question is, can we move from kill everything to precision control? Can we actually use bacteriophages, these viruses that hunt bacteria, to surgically remove contaminants from our bioprocesses without harming the product?
It's a seductive concept, I'll give you that. You're proposing we introduce a predator to guard the flock. But I'm here to pump the brakes a little. You're talking about intentionally introducing a self-replicating biological agent into a system that relies on absolute predictability. I think you're trading a chemical engineering problem for a chaotic, evolutionary one. And I'm just not convinced the risk profile makes sense for high-stakes manufacturing.
And that really is the core of the disagreement, isn't it? My view is that industrial phage therapy is a necessary evolution. Our traditional antibiotics are failing because of resistance, and chemical sterilization is just a blunt instrument. It damages equipment, it's bad for the environment. I believe engineered phages offer a surgical, self-amplifying solution that we can't afford to ignore anymore.
And I take the position that while the biology is, well, elegant, the operational reality is a minefield. I think the speed of bacterial evolution, specifically resistance, and the sheer risk of introducing viruses into a plant create a high-wire act that most manufacturers simply can't afford to try.
All right, so let's start with the mechanism, because I think elegant is exactly the right word. The current tool set it's obsolete. We rely on these broad-spectrum antibiotics or aggressive heat, and these have three massive flaws. They create resistance, they can damage the sensitive proteins we're actually trying to make, and they are environmentally hostile. This is where the phage comes in. It's nature's sniper. The mechanism is a lock-in key. A bacteriophage targets specific surface receptors on the contaminant, say an unwanted E. coli or bacillus, and it completely ignores the valuable production strain.
I don't dispute the specificity mechanism in a vacuum, no.
But here's the real differentiator. And this is why I am so convinced this is the future. Phages are self-amplifying. Unlike, say, a chemical sanitizer that starts diluting the moment you add it to the tank, a phage replicates. As long as that contaminant is present, the phage infection cycle continues, producing more and more phages to hunt down the remaining bacteria. It's a dose-responsive defense system that scales with the threat.
I see why you think that. And on paper, self-amplifying sounds like a dream. But let me offer a different perspective. That same specificity you're calling a feature, I call it a massive vulnerability, maybe even a fatal flaw. Because phages are so specific, often down to the strain level, they are incredibly fragile tools. If that contaminant has a single-point mutation in its surface receptor, just a tiny genetic shift, the phage is useless. It's like the lock just changed the keyhole.
But that assumes a static situation.
It assumes the reality of evolution. I mean, bacteria have been fighting off phages for what, 4 billion years? They have robust defense systems, restriction modification systems that literally chop up viral DNA. They have CRISPR-Cas, which is effectively a bacterial immune system. The moment you introduce a precision agent, you are kicking off an arms race right there inside your bioreactor.
Okay, that's a fair point, but I would frame it differently. You're describing the limitations of wild-type phages. You know, the stuff we just pull out of a sewage sample and dump in a tank. That isn't what we are proposing here. We're talking about engineered precision. We've moved way past the era of just seeing what we can find in a ditch.
Even with engineering, you're chasing a moving target.
But we have the blueprints now. We can sequence phage genomes completely, and this allows us to strip out the dangerous parts. We can identify and remove virulence factors, toxins, or any genes that might lead to lysogeny, where the virus just goes dormant instead of actually killing the host. We are designing strictly lytic phages. And beyond that, we're looking at things like endolysins.
Right, but endolysins are enzymes, not the virus itself.
Correct, but they're derived from the phage mechanism. Think about biofilms. You know those bacterial fortresses that form in the dead legs of piping where steam just can't reach? Chemical cleaners, they just bounce right off that extracellular matrix. But engineered phages can express depolymerases, enzymes specifically designed to chew through that slime. That's a capability chemical engineering just doesn't have.
Okay, I'll grant you that biofilms are a massive headache and enzymatic cleaning has potential. But let's go back to the phage itself. I'm sorry, I just don't buy that we can engineer our way out of evolution so easily. If you use a monoclonal therapy, a single type of phage, the bacteria will evolve resistance in a heartbeat. The source material is very clear on this.
Which is precisely why we don't use single phages. We use cocktails.
Right, cocktails. The standard answer. But have you really considered the complexity of designing a stable cocktail? You can't just throw 5 phages in a bucket and hope they work together. Phages have antagonistic interactions.
We can screen for that, though.
It's harder than it sounds. There is this phenomenon called superinfection exclusion. If phage A gets into the cell, it starts producing proteins that actually block phage B from getting in. They compete, so you might think you have a multi-pronged attack, but inside the tank, your phages are just fighting each other for receptors. You are trying to engineer a stable ecosystem inside a bioreactor that is already a high-stress environment. And even if you design the perfect cocktail, that genetic blueprint you mentioned only works until the bacteria mutates. Then you're back to square one, needing to update the cocktail. It's a never-ending game of catch-up.
I think you're underestimating the speed at which we can now characterize and adapt these cocktails. High-throughput screening lets us test huge phage libraries against panels of bacterial isolates very, very quickly. We can rationalize the selection to avoid antagonism, but I want to pivot back to something deeper here. You mentioned the operational risk.
The friend and foe problem, yeah.
Exactly. You're worried about the phage attacking the production strain.
Absolutely. I mean, in many fermentation industries, like dairy, for example, phages are the enemy. They're the things that destroy the starter cultures. So you're asking facility managers who have spent their entire careers terrified of phage contamination to now deliberately introduce phages into their pipes? It creates a landscape that is just incredibly difficult to manage. You are risking catastrophic lysis of your production host if you get the engineering even slightly wrong.
And that is a valid concern, but the specificity is our shield there. We can screen for cross-reactivity with the production strain during development. It's a standard QC step. But more importantly, look at where this is already working. In the food industry, phages are used to reduce listeria and salmonella on meats and cheeses. They reduce pathogen counts by multiple logs without harming the product quality at all.
A surface treatment on a wheel of cheese is physically very different from a submerged bioreactor.
True, but the biological principle holds. And we have data from environmental biotech, specifically ethanol fermentation. They have successfully used phage cocktails to restore yield in batches that were contaminated with lactobacillus. They didn't just sterilize the tank, they saved the batch. That is yield stabilization. Instead of dumping the product, you suppress the contaminant just enough to let the production strain finish its job. That is millions of dollars saved.
That's a compelling argument, but let's look at the kinetics. You call it yield stabilization, I call it a timing gamble. There's a concept called kinetic lag.
Go on.
In a nutrient-rich bioreactor, bacteria grow explosively. They can double every 20 minutes. Phage replication, it takes time. There's a latency period, a burst size. If the phage replication lags behind the bacterial bloom, even by a few hours, the damage is already done. The metabolites are ruined, the nutrients are gone. The phage might win eventually, sure, but it wins the battle for a batch that's already dead.
Well, that's why early detection is key. We aren't waiting for the tank to fail.
Even with early detection, you have an environmental mismatch. A phage that works beautifully in a petri dish at neutral pH might fail miserably in a bioreactor with high solids content, variable pH, or just the immense shear stress from an industrial impeller mixing at high speeds. Phages are physical structures. They have these protein tails. They can be literally mechanically destroyed by the mixer.
Shear sensitivity is a solvable engineering parameter. We can formulate phages with excipients or even encapsulate them to survive the dosing process. But I want to look forward a bit. You seem focused on the difficulties of today, but I am looking at the cyber-physical future of biomanufacturing.
Cyber-physical, that sounds like a buzzword.
It is the integration of biology and data. Imagine a system where sensors detect the earliest metabolic signatures of contamination, a shift in off-gas profiles maybe, or a tiny change in turbidity. An AI algorithm analyzes this, identifies the likely contaminant, and then triggers an automated dosing of a specific phage cocktail tailored to that very signature. It transforms phage therapy from a static addition to a dynamic feedback loop. It's closing the loop on contamination control.
I come at it from a different way. I see that level of automation as just multiplying the points of failure. But let's talk about the aftermath. Let's say your AI works, the phage works, the batch is saved. What happens to the phage?
Well, ideally, it's removed during downstream processing.
Ideally, but regulators are going to scrutinize phage fate intensely. Where do these viruses go in the waste stream? If you're discharging thousands of liters of genetically engineered, or at least industrially selected, phages into the wastewater, what's the environmental impact?
They're biodegradable. They're just proteins in DNA. They break down.
But before they break down, they are vectors. And here's the really scary part: horizontal gene transfer.
That's a risk in any biological system.
But phages are the primary mechanism for it. It's called transduction. Phages can accidentally package bacterial DNA, including antibiotic resistance genes or virulence factors, and shuttle them between bacteria. By flooding a system with phages, you might actually be accelerating the spread of undesirable genes. You could be turning your wastewater treatment plant into a breeding ground for superbugs.
Transduction is a known phenomenon, but we can screen for generalized transducing phages and exclude them. We can select phages that have degraded packaging mechanisms for host DNA.
You can try, but biology is messy. And frankly, the regulatory pathways for this are murky at best. How do you validate a living cleaning agent that changes over time? And there's another hurdle we haven't even touched on: the manufacturing burden.
What do you mean by burden?
I mean that to use phage therapy, you essentially have to become a phage manufacturer. You have to propagate them, clarify them, and, this is the hard part, remove the endotoxins. If you're growing phages on E. coli, the solution is full of toxic cell debris. If you're making an injectable pharmaceutical, you cannot introduce a cleaning agent that is itself full of endotoxins. You are adding a massive layer of complexity and cost to your supply chain just to have this backup plan.
I'm not convinced by that line of reasoning, because the alternative is often losing the entire batch or shutting down for days to decontaminate. The cost of the phage preparation, even a high-purity one, is negligible compared to the cost of a lost API campaign. It's an insurance policy. And regarding the environmental mismatch, yes, pH and shear are factors, but that's what stability testing is for. But let's look at the bigger picture here: sustainability.
Sustainability is important. I agree.
It's critical. The industry is under immense pressure to reduce its chemical load and its carbon footprint. Phages allow us to reduce reliance on aggressive sanitizers, on sodium hydroxide, on thermal sterilization, which burns massive amounts of energy. It is a green processing tool. It aligns with where the world is going.
I agree that the sustainability narrative is strong, but we have to ask if the complexity justifies the gain.
You are talking about a system that requires constant surveillance, constant updating of cocktails, and rigorous containment to prevent the friend from becoming a foe to the main production line. If you're using E. coli to produce insulin and you have an E. coli phage on site for cleaning, a simple leak in a valve could result in that phage wiping out your production line. That is a catastrophic failure mode that just doesn't exist with bleach.
But that's a risk of containment, not the technology itself. We handle dangerous organisms and potent compounds all the time. Facility design can segregate these flows. We can distinguish between the tool phage and the threat phage.
It requires a level of operational perfection that's, well, it's hard to maintain in the real world. Valves leak, humans make mistakes.
Perhaps. But the alternative is the status quo, which is failing. We cannot keep relying on total sterility myths. The data shows that broad-spectrum approaches are losing efficacy. We need a tool that can selectively sculpt the microbial community. Even if it requires complex engineering, the ability to save a campaign that represents months of work and millions of dollars is worth the effort to solve these engineering hurdles.
We are nearing the end of our time, so let me just summarize my position. I think industrial phage therapy is a fascinating inversion of control, using a predator rather than a poison, but we can't gloss over the fact that it transforms a chemical engineering problem into a complex ecological one. Success hinges on managing an arms race between phage and bacteria inside a tank that is critical for business. You are betting that you can out-engineer 4 billion years of bacterial evolution, and I'm just not sure the house odds are in your favor.
And I will leave you with this. The industry cannot afford to stand still. We are facing a future where sterility is harder to guarantee and our traditional tools are being regulated away or just rendered useless by resistance. By moving toward the selective sculpting of microbial communities, we gain a tool for resilience. It isn't easy, and it requires deep scientific rigor, but it turns a binary pass-fail system into one where we can actively defend our processes. The invisible enemy is evolving. Our defense has to evolve with it.
It seems we do agree that the future involves managing ecosystems, not just sterilizing vessels.
Absolutely. The days of simply boiling everything to death may be numbered.
So we invite you, the listener, to consider, would you trust a virus to keep your medicine pure?
Thanks for listening to the debate.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.